All right. Good. Hey, everybody. Let's pick up where we left off with animals last time. We had gotten through talking about tissues and what becomes of them and so on, and now we are going to talk about another developmental change during the early embryonic stages, and that is the development of what are called body cavities. So let's not delay and okay. A body cavity is a space where the organs exist, like your liver and your heart and your lungs and your stomach. Your body has two cavities in it. Okay, it has the inside of your stomach where your food gets digested. Okay, and then it's got another space in which all of your organs exist. That's what the space we're talking about when we talk about a coelom, not your inside of your stomach, but the body, the place in your body where you would open up and see all of your organs. Okay. So a fluid-filled space that separates the digestive tract from the outer body wall. It's a space that contains the internal organs. Okay. All right. Why have one? It is, first and foremost, it's difficult for organs to develop if there is not a space in which for them to do so. So, it simply allows this development of the body organs as separate entities in and of themselves. But it is a fluid-filled cavity which allows the organs to be protected and cushioned. In soft-bodied animals, it's this fluid gives them, when they don't have a skeleton, like a worm, for example, it gives them still something that their muscles can work against and use it to move their bodies. It's referred to as a hydrostatic skeleton, but it's just the fluid. Organs can move independently of the outer surface, which if you think of your heart being able to beat, but your outside of your body is not moving with that, that's kind of what we're talking about. Now, some animals, as fundamental as a body cavity may seem to us, some animals lack them completely. And some animals, like us, have a true body cavity, and some animals have a sort of quasi one. So we'll talk about these. First of all, acoelomate animals. Acoelomate means without. So these are the animals that lack a coelom. Okay? They lack a body cavity. I want you to think flatworms, though there are others, but this is a great example of one. This is a poor little planarian flatworm that has been cut in half. These are guys you might have seen in your first year of biology. They're like 2 centimeters long. And you notice if you cut this guy across his middle, what you see here inside this hole, this tube that would run the length of his body, that is his gut. That's where he digests his food, okay? And this is that endoderm, and you can see that it's completely surrounding his gut cavity. This in the red is mesoderm tissue, and this in the blue is ectoderm tissue. But he really has no I won't say no, but no well-developed organs. He's got the beginnings of some organs, but none that are really well-developed. He does not have a body cavity in which to place them, okay? So they have a gut, but for food, but not a hollow space to contain the organs. He is a solid-bodied critter. His body is solid except for that gut space. Okay. Now, Pseudocoelomate animals. In order to understand what these are, you have to understand one more thing about a coelom. A true coelom is a space that has mesoderm completely surrounding it. So it is lined completely with mesoderm. Okay? Now, pseudocoelomate animals have what, for all intents and purposes, looks like a coelom. Okay? It is a body space. Organs could develop there, but it is not completely lined with mesoderm. And so it does not meet the definition of a true coelom. Here you have a roundworm. These are like nematodes. Roundworms are extraordinarily common worms, and they're everywhere in the environment, but sometimes they can be parasitic. Many times they are not. But if we cut this guy across like that, he has this mouth up here and a gut that goes all the way through his body down to his anus, and so that is showing you the cross section of his gut tube, and there you can see the gut right there, completely surrounded with endoderm, and that's where his food would be digested. Now, if he were truly a coelomate animal, you would see a ring of red around that endoderm. Then you would see this space, the coelom, and then you'd see this other ring that you do see around here of mesoderm. So you would have mesoderm, space, mesoderm. In this animal, you don't have that. You just have the endoderm, a space, and then mesoderm. So it is a pseudocoelom. It looks like a coelom. It sort of acts like one. He doesn't have a whole lot of internal organs other than some reproductive ones, but generally speaking, that's what a pseudocoelom, and it's not a very common structure among animals, but that's what it means. Excuse me. All right. Now, the coelomate animals have a true body cavity, and it is completely lined with mesoderm. And so here you see it. This is an earthworm. It is a truly coelomate animal, and you can see again, here we cut him across. We are looking in the tube that is his gut. There's where his digestion would occur. The endoderm surrounds that cavity, and then mesoderm surrounds that endoderm. There is a space, and then there is more mesoderm and ectoderm immediately attached. Okay, so that's what we are talking about when we're talking about a true fluid-filled body cavity housing body organs. The earthworm is where we really do start to see some sophistication in organ structure and so on. All right, now these animals that have a true body cavity, have a true coelom, can be divided into two groups based on how they develop as embryos. These two groups are the protostome group and the deuterostome group. Okay, and these two types of development are distinguished by three different features: how the cells cleave from zygote as we're cleaving to form the blastula, so how they cleave, how their coelom forms, and what is going to happen to a little structure called a blastopore, and we will talk about that. So, protostome animals and deuterostome animals. First with the cleavage. Protostome animals show what is called spiral cleavage, where instead of what we saw earlier, instead of this neat, where can I draw this? I'll draw it over here. Instead of this neat radial perpendicular line cleavage that goes right down the middle of the organism, it tends to shift around and create a spiral pattern. Okay? Sorry, folks. So this is showing you a comparison of that. Deuterostome animals have the radial. We are deuterostomes, so that's the one that you probably need to pay the most attention to. The spiral is for other critters, and we'll talk about who's got this stuff in a minute. Determinate cleavage is what we see in protostomes. Now, what this means is that already, even at this early stage of cleavage, these cells have a definite fate. They are destined to become certain types of tissue or certain body organs. So if you plucked one of those cells off of this embryo over here, okay, it could not develop into another embryo, and this embryo that you plucked it from would be truly missing something, okay? Whereas indeterminate cleavage, which is what we see in this type of division, in deuterostomes, if you plucked this cell off of this ball of cells, it could become a whole nother organism. At this stage, still, it retains the ability to do that. And in fact, if you plucked the cell off, well, the remainder would still become an intact other organism. So each embryonic cell retains the ability to become a new embryo in this case. This is what makes identical twins possible. This zygote, or this little ball of cells, can split, and each of them can become an identical twin. That does not happen in spiral cleavage. It cannot happen in spiral cleavage. Okay? So it makes the embryonic cells extremely important, possibly in being able to cultivate tissue types because they still are very versatile, and this leads to the controversy surrounding stem cells, and maybe we can talk a smidge about that. We need now to talk about the formation of the body cavity. Okay, and how that ties into the fate of the blastopore. Now, when you were a little blastula, let me get my pen back. When you were a blastula, you turned yourself inside out. So you were a hollow ball of cells, and you turned yourself inside out. Okay, and when you did so, the place where you began doing that remained as a little hole. This is called the blastopore. Okay? And that opening is pretty important. Okay? It is leading to that archenteron we talked about, that gut cavity. So it is the opening that leads to the gut, that archenteron space, which means we have two openings that we're aware of to our gut cavity, our mouth and the other one, okay? Mouth to anus with the tube in between. Well, here we see one opening, and if you are a protostome, this opening is going to become your mouth. If you are a deuterostome and follow this kind of development, that blastopore is going to become your anus. It is a fundamentally different way to look at the world. Okay, the blastopore and the second, yes, this is what I just told you. Okay, so once the second opening develops up here, okay, you've got the beginnings of that complete tube. But the fate of this blastopore is one of the things that determines whether you are a protostome or a deuterostome. Okay? Now, in a protostome, as I just said, this is an important thing to keep in mind. The blastopore is going to become the mouth. Now, who follows this type of development? Segmented worms do. Its blastopore becomes its mouth. Mollusks like squid and snails and things, and all arthropods, everything from crustaceans, crabs, to insects, grasshoppers, all of that. So these are basically almost all of your invertebrate animals are protostomes, okay, except for the ones that are so primitive they can't even have a coelom, so we can't have this discussion, okay? Now, deuterostomes, the blastopore becomes the anus. Who does this? Basically two major phyla. Echinoderms, who are starfish, and if you look at those, you're like, whoa, they're pretty primitive. They don't even have bilateral symmetry, but we need to talk about that. And then all of us vertebrates, all vertebrates, all chordate vertebrates, well, there's no other kind of vertebrate, but have this deuterostomic blastopore becomes the anus type of development. So what this means is that this guy, though he looks really much less like us than, say, a lobster does, at least a lobster's got bilateral symmetry and so on, this guy is our closest invertebrate relative. So if you're on the beach and you see a starfish, you can say, howdy cousin, because he is your closest invertebrate relative. This is what he looks like when he is a larva, and this is what links him to us much more than his adult form. We see a bilateral type symmetry. We see the formation of the gut. Okay? All right. Last thing to talk about. There are about 35 animal phyla. Remember, phylum is the next level down from kingdom. There's about 35 of them. We aren't going to learn near that many. We'll talk about maybe about 8 or 9 of them. And of course, like all things taxonomic, the relationships between them are somewhat in debate right now. It's a very active field. A lot is being revealed through actually analyzing the genes of different organisms to see who's related to whom. But there are certain points that are in pretty much agreement. First of all, the major point is that all animals do share a common ancestor that scientists think probably looked like that choanoflagellate we talked about, but it's believed that it's a monophyletic group, meaning all animals originated from one common ancestor. Sponges are at the base of it. They are the most primitive, and this says it is a paraphyletic group, meaning they need to still work on the sponge classification. They really think they've got a bunch of different phyla still stuck in one group, and they haven't sorted it out yet. So that's what they mean by paraphyletic. Now, the eumetazoa that you see here is just the term given to all animals that have true tissues. Remember, what separates sponges from the rest of the animals is their very primitive nature. They don't have true tissues. They're almost colonial. They're so primitive. So the step from sponges that's more advanced are all the animals with true tissues. And at the base of that group of animals, the cnidarians, or the jellyfishes and the sea anemones and those kinds, they are the ones that are at the base of that. So sponges are at the base of all of it, but for the vast majority of animals, the cnidarians are the next step. Okay? Most animal phyla are bilateral in symmetry, so they belong to the bilateral group. This term clade is just the term for the part of a family tree where you have one common ancestor and all its descendants. So the bilateral group is one group where it's believed there is a common ancestor that gave rise to all of them as well. So, vertebrates and the echinoderms belong to the deuterostome. So let's see how this would all play out. All right, you've got some ancestral choanoflagellate type protist thing that doesn't meet the criteria for truly being an animal, but there it is. And this is the first split. So this common ancestor gives rise to all the sponges here, and that is phylum Porifera for us today. And it's, again, thought this phylum probably needs some sorting out still. Then we've got this branch over here that leads to all of those animals that do have at least true tissues. So that's the first separation. No true tissues, true tissues. Okay? Now, there was a common ancestor represented at this nexus right here. That then gave rise to all the bilaterally symmetrical organisms. Okay, so our primitive ones that have just the tissue level of organization remain here in these two phyla today, Cnidaria and this group, which we won't talk about too much, but it's another type of jellyfish creature, but they are so distantly related. Their last common ancestor is way back here. Even though they look very similar, they're only distantly related. Now, if we follow this path, there is a common ancestor right here that then gives rise to this more advanced group. So the ancestral group looked probably the most like these with no coelom, which is only our flatworms, our Platyhelminthes group that still exists today. But the more advanced group, this in here, some acoelomate animal at this point gave rise to animals that actually had body cavities. Okay, and by that we mean any type of coelom, true or pseudo. So right here, a common ancestor then gave rise to these groups that have a true body cavity, and within that group there is the sort that has the deuterostomic development and the protostomic development. And so those are the basic splits. We can see the protostomes include the mollusks, the annelids, and the arthropods, and the deuterostomes, the echinoderms, and the chordates. And this shows that they share a recent common ancestor, or more recent, than either of them do with this group over here. And these groups are more closely related to each other. And these groups here are more closely related to each other than either of them is to the mollusk group. Okay? And that makes sense, because if you look at this group, the earthworms and the arthropods, they both have body segments and so on, so we see that commonality. All right, this is just another image showing essentially the same thing. I think we're done. So, if you've got questions, let me know. Otherwise, we're done with the introduction to animals, and now we can start focusing on their organ systems. Thanks a lot, and I'll see you next time.